Articles and MCQs. Guys, our today's topic of discussion is unit membrane model of plasma membrane. Guys, you know that the plasma membrane, or that is simply called as cell membrane, also called as cell membrane, is the outermost boundary of the cell. Means if we look at this cell, then the outermost boundary or outermost wall of the cell is called as cell membrane or plasma membrane, and it is the outermost boundary in the animal cell, but in the plant cell it is also covered by another wall that is called as cell wall. Guys, today we will discuss the complete structure of this cell membrane. But before starting the structure, let me tell you about one thing, that the plasma membrane is facing the cytoplasm in the inner side, and on the outer side of the plasma membrane or the cell membrane is the extracellular fluid of the cell, means the fluid that is present outside the cell. So, when we will discuss the structure of the cell membrane, these two things are important. That on the outer side, extracellular fluid is present, and on the inner side of the plasma membrane, it is bounded by cytoplasm, or simply that is called as cytosol. And if we talk about its chemical composition, we will see that the plasma membrane consists of the proteins, lipids, and carbohydrates. These are the main things that are present in the plasma membrane. And the proteins, the percentage of the proteins is 60 to 80%, while the lipids are about 20 to 40%, and the carbohydrates are in very small quantity present present in the plasma membrane. So, many biologists, guys, contribute to establish the structural organization of the plasma membrane, and they designed the two models. The first one is a unit membrane model, and the second one is fluid mosaic model. But in this lecture, we will discuss about the unit membrane model, and we will discuss the fluid mosaic model next. First of all, the unit membrane model was proposed by Danielle and Davson in 1935. And it was the first model of the plasma membrane that was proposed, and it was not that accurate. But the fluid mosaic model was accurate. Let us discuss that what is this model. Guys, according to this model, there are the two layers of the lipid are present. Here you can see that this is the lipid layer. This is the first layer of lipid, and this is the second layer of lipid. So these are collectively called as lipid bilayer. And in the outer portion of this lipid are the layers of protein in this portion, and this is that portion, means this is the outer portion of the plasma membrane facing towards the extracellular fluid, and extracellular fluid is in direct contact with this protein layer, while in the inner side of the plasma membrane, that is towards the cytosol or cytoplasm, it is also in the direct contact with the proteins. And neither cytosol and nor extracellular fluid is in contact with the lipid layer. These both are in direct contact with the protein layers. So we can say that the lipid bilayer is sandwiched between the two protein layers, because it is like a sandwich, that the two protein layers are present on the outside and the lipid bilayer is present on the inside. So it is called as unit membrane model. According to this model, guys, there is the hydrophilic zone that is present outside, and hydrophilic zone is made up of this protein layer and the head region of the lipid layer. This circular region is the head region of the lipid layer, and these thread-like structures are called as tail, and these tails are called as hydrophobic zone. Hydrophilic means water-loving. The proteins and the head region of the lipid is water-loving, and the tail region of the lipids are hydrophobic, means they are water-repelling. So these main are the two zones, hydrophilic zone and hydrophobic zone. Guys, if we talk about that which type of the proteins are present in the plasma membrane, then this is a globular proteins. While if we talk about the lipid bilayer, then the lipid bilayer consists of the main lipids that are called as phospholipids. So this is all about the unit membrane model of Danielle and Davson, which was proposed in 1935. But as it was not accurate, so the next unit membrane model was proposed by Robertson, and it was proposed in 1959. And it was not also so accurate as compared to the fluid mosaic model, which we will discuss in the next lecture. This Robertson model is almost same as the Davson and Daniel experiment model. And according to this model, the plasma membrane also consists of the lipid bilayer, and these are the lipid bilayers. And same is the case the head regions are the hydrophilic regions of the lipid bilayer, and the tail regions are the hydrophobic regions of the lipid bilayer. So these yellow portions are hydrophilic and these purple one are hydrophobic. But what is the difference between the Davson, Daniel and Robertson experiment? It is on the basis of the layers of protein molecule. It is the outer protein layer that is towards the extracellular fluid, and it is the inner protein layer that is towards the cytoplasm. You can see that both the structures of the protein are drawn differently. They are based upon the difference in the type of the outer and inner protein layer. So this is the minor difference, that according to the previous model, the protein layers, the type of the protein layers were same. But according to the Robertson model, the outer and inner protein layers, the type of the outer and inner protein layers are different from each other, and so their structures. So this is the all about the unit membrane model of the plasma membrane. And one more thing, that the model of the Daniel and Davson is also called as sandwich model due to its structure. But the Robertson model is not called as sandwich model. It is simply called as a unit membrane model of Robertson. So guys, I hope you understand all the things about this lecture, and don't forget to share this video with your friends. Thank you so much.